don't know flying where, but okay. So let's first look at the misconception and errors, common errors among the student in all the chapters, okay? So the first chapter here, so you stop me to explain if you wanna, or else I'll quickly go through with you, okay? Shall I make it bigger so you can read? Okay. Right, so I'll go through very quickly because there's so many chapters, right? So for cell structure, common mistake, measuring images or specimens in cm and forgetting to times 10 to get mm. So by now you already know that every time you want to measure something using your ruler, the unit must be in mm. Right? So therefore, when you're asked to do calculation, when you make your measurement and write down your measurement from your ruler, please write it down in mm. Because if you look at the mark scheme, it's also in mm. Some students write in cm, I also accept, but then they said you must, you see, they said forgetting to times 10 to mm means they want mm, they don't want cm. Strictly speaking, if you write cm, you don't convert to mm, they can refuse to give you mark. You see, before converting to micrometer. Okay, that means if you measure in cm and straight away you convert to micrometer, strictly speaking, you cannot give, I cannot give mark in your trial exam, right? So make sure you know this. Next, stating that the nucleus is surrounded by a nuclear membrane. So nucleus is double membrane, right? So the membrane itself is actually 2 layers of membrane, and the outer inner layer will form the nuclear envelope. So the envelope is double membrane, okay? Stating that lysosome digests bacteria rather than enzyme they contain. So lysosome is an organelle, it cannot digest. The enzyme, the hydrolytic enzyme is the one that digests. So from this, you know that how detailed you have learned in A level is to that detail you need to write your answer so that your answer is accurate and therefore qualify to get the mark. Okay, so you can see that. Stating that naked DNA, so what's the meaning of naked DNA? Naked DNA means there is no histone protein. It doesn't surround the histone protein. So this is the meaning of naked DNA. Okay, it doesn't mean that it is not surrounded by nuclear envelope. So please, when you look at the name, you need to learn up the meaning. Believing that viruses have cell walls. Virus got no cell wall. Virus got protein code. Okay, so this is for chapter 1. Now let's go to chapter 2, biological molecule, common mistakes among student exam. Using the term iodine. So I already said cannot use iodine because iodine is a purple color gas, the I2. I2 is a purple color gas, so you cannot write iodine. You need to write, okay, if you say you want to stain whatever the starch with iodine, you must say stain with iodine in potassium iodide solution or I in Ki, because iodine can be I2. I2 is a purple color gas. So that is the meaning of you need to write your answer accurately to that detail you have learned, to that detail to write, okay? Confusing the roles of Benedict's reagent with Buret solution. So this one, you know, Benedict is for reducing sugar test. Benedict is not for glucose, Benedict is for reducing sugar test, okay? Buret is for protein, okay? Missing out the H on carbon 5 when drawing the ring structure of alpha and beta glucose, or writing C2H5OH instead of CH2OH, missing out the H on carbon number 5. So this is, let's say, glucose, right? Okay, it's a 6th carbon, whereby the 5th carbon is outside the ring because one of the corner is occupied by O in the ring. So therefore, the last carbon is outside here. So this is actually CH2OH over here because there's a carbon, right? So carbon then have got 4 covalent bond. So you've got 1 OH, isn't it? So when you've got 1 OH, the other one must be hydrogen and hydrogen. So CH2OH. So you can write here CH2OH when you're asked to draw the structure. State that denaturation of protein is because peptide bonds or disulfide bonds are broken. No, when the protein is denatured because the cross links between the R groups are broken, you know, the tertiary structure protein got a lot of cross links, right? The 4 types of cross links. So when the cross link the cross links, the 4 types of cross link or a few types of the cross links are broken, or one type of cross link is broken, then the protein will change shape. So what are the 4 types of crosslinks that can be formed? Aaron, 4 types of crosslinks between the R groups. What are they? You can unmute to give the answer, or you can type. So stating that water has specific heat capacity and latent heat of vaporization, forgetting to use the term high. So when you learn the name of a certain property, you need to give the full name. You cannot simply change the name, like I cannot simply change your name and call you something else, right? So a name is a name, whether given to a structure, given to a process, you must use the full name. High specific heat capacity is different from specific heat capacity. Okay, so this common mistake. So number 3, enzymes. Using the term extrinsic or exocellular for extracellular enzyme. So when you learn the term as extracellular enzyme, use back this term is actually a name. Don't simply interchange with something else like extrinsic, exocellular, because this is not the name given to this enzyme that is secreted out of the cell to be used outside the cell. It's called extracellular. So don't simply change the name, okay, of a biological term or a structure or a process. Okay, stating that enzymes bind substrate, forgetting to use the term active site. So now you know that substrate must always sit into the enzyme active site here. So this is the substrate, right? So in A level, you already learned substrate must always sit into the enzyme active site. So if you say enzyme bind to substrate or substrate bind to enzyme, it is not specific enough to the detail that you have learned in A level, you lose mark. So to that detail you learn, to that detail you write an answer to score full marks for every question. Full marks. It is possible. Okay, this is how you score full marks lah. Right? Stating active site. Oh my God, what is active site? Active site, the spelling also, see not? Okay. Believing that the Michaelis-Menten constant is a rate value rather than a substrate concentration. Michaelis-Menten constant is Km value, right? So Km value is found on the graph, isn't it? Of rate of reaction versus what? Annabelle, rate of reaction versus what? Annabelle? Hurry up, I'm recording. So substrate concentration. So when you find your Km value, isn't it? You look at the Vmax, and then from the Vmax, you look at the half Vmax, right? This is half Vmax. Vmax, I have to write max, divided by 2, half Vmax. And then you interpolate to this. This on the x-axis is substrate concentration. This is the Km value. So the Km value is actually the substrate concentration, not the rate. Okay, so it looks like this is common mistake among the students. Next, okay, cell membranes and transport. Stating that cholesterol, okay, stating that cholesterol has a hydrophilic head and a hydrophobic tail. What's that? Hydrophilic head and hydrophobic tail. Cholesterol. No, we never learned that cholesterol got this thing. We only learned the phospholipid molecule has. So don't simply cut and paste, okay, in A level. Okay, must be specific to what you learn only, right? Confusing channel and carrier protein. This one everyone know already, but it's channel protein, it's carrier protein already, right? So channel protein is drawn like this one. It's a little river, it's very small. Only small ions can pass through. Carrier protein is a big bag, right? You can carry glucose, all those big molecules, okay, that are polar, right? Confusing cell surface receptors and cell surface antigen different, right? Cell surface receptor is supposed to actually allow a signal molecule to bind, isn't it? Okay, whereas cell surface antigen is actually, it can be a glycoprotein or it can be a glycolipid, which act as an antigen, isn't it? Because you got this glyco means you got this carbohydrate chain on protein, carbohydrate chain on phospholipid. So this This is your surface antigen, these two, okay, which is different from your receptor. What is the molecular structure of receptor?
Is it a protein?
Okay, glycoproteins and glycolipids are both of them can be the receptor site for chemical signaling, can also be antigen. So that means the nature of the receptor and antigen molecule can be glycoprotein or glycolipid. But antigen is antigen, receptor is receptor. They are different, but the nature of the molecule that form them are the same. Okay. Stating that cell signals start off a nerve impulse. No, the cell signal that bind to the receptor will cause the 5 steps in cell signaling, right? The transduction and the response. So the 5 steps in cell signaling you need to remember. Every time they talk about cell signaling, you go back to your AS syllabus. What exactly about the cell signaling you're supposed to learn? Those are the things they're supposed to be mentioning. So these are the 5 things in your cell signaling that you need to remember. The 5 things, these 5 things here, right? So you got secretion, okay, from the endocrine gland cell. Then you have got transportation in the bloodstream. Reception is where the hormone bind to the receptor. The hormone is first messenger, extracellular messenger signal molecule. Then transduction number 4 is when you have got the second messenger that's formed inside the cell. So it's called intracellular messenger or intracellular signal molecule. Then activate a series of cascading reactions to amplify the signal. So transduction, it got 2 parts, secondary messenger and also cascading reactions of the enzyme. Then lastly is the response, means what does the cell do because of receiving this signal. So these are the 5 things in your syllabus that you need to know and you refer to as your base to answer question related to cell signaling. Okay, so always go back to what is it you have learned. That's what you need to write. Very easy. What they want the mark from is what you have learned. Okay. Stating that facilitated diffusion requires energy, that it is movement against concentration gradient. So that means a student confusing between facilitated diffusion versus active transport. Facilitated means you need something to help you to transport across the membrane. So you need a carrier protein or channel protein, right? So this is called facilitated. Whereas diffusion means you don't need anything to help. It can just diffuse by itself across the gap between the phospholipid molecules. Okay. Using the term concentration of water. So yeah, this is, I keep on stressing, water has no concentration one. Water got no concentration, only solution got concentration. Right? Water is a solvent. How can it have concentration? So if you want to say the amount of water molecules, right, then you have to say water potential. Water potential will tell you the rough amount or number of free water molecules that are not attached to any other solute. They're free to move. The more you have, the more they can move. So you need to learn up the actual meaning of water potential. Don't use concentration for water. Okay. Mitotic cell cycle, confusing between chromatids and chromosomes. So now you know it's chromatids and chromosome, right? Chromatids is one of the 2 copies of the DNA produced by DNA replication, right? Sister chromatids, right? Chromosome is that DNA that wrap around the histone protein, is called a chromosome, okay? It will condense and thicken during mitosis. So this is definition of biological term you have to learn up. Why? What happens if they ask you, okay, give the definition of chromatids versus chromosome in the exam? 2 marks. Can you score the 2 marks or not? You must make sure you can score these 2 marks because every question you want to try to score full marks. Okay, full marks. So confusing centromeres, centrosomes, and centrioles. Okay, what's centromere, centrosome, centriole? Oh, you know or not? Okay, Losician, what is centromere? Losician, what is centromere?
Centromere is the held between the chromosome held between the sister chromatids.
So it's the point where the sister chromatids are held together, right?
Yeah.
Okay, good. Okay, or attached together in the chromosome. Centrosome. What is centrosome? Huh? Okay, what is centrosome? Timing jet. Okay, timing jet. What is centrosome? What is centrosome?
An organelle that synthesizes no fiber.
Organelle, where's location? Location where? Function? Okay, organelle, very good. It's an organelle. Good, 1 mark. Location where? Sarah, location of, okay, thank you, Minjet. Sarah, location centrosome where? Faster, faster.
Is the centrosome like the microtubule organizing center?
Yes, so you're telling me the function, right? I'm asking you where. Good, function is MTOC, microtubule organizing center. That means this is a organelle, which is actually a location in the cell where the microtubules are being synthesized, right? Microtubules of the cells are being synthesized so that you can synthesize the structures that need microtubules, like centrioles, like cilia, like flagella, right? Okay, MTOC, very good. So where's the location of this centrosome? Centrosome is also where centrioles are found, you know. So where centrioles are found? Where is it found? Ryan? Ryan.
Is it cytoplasm?
Yes, cytoplasm. Exactly like where? Is it near the nucleus or near the cell surface membrane?
Nucleus.
Yes, all the centrioles are near the nucleus, isn't it? It's very near to nucleus. So therefore, if you can find that centriole, so this is the region where the centrosome will be. Why is good to learn today? Just in case they draw a cell and ask you to label the location of centrosome, you know. Can you label or not? Okay, next question. Stating that, okay, thank you. Stating that mitosis is for cell repair. Mitosis is for cell repair instead of tissue repair. This is a common mistake among students. Mitosis does not repair the cell. When the cell already damaged, right? You destroy the cell. Why you go and repair the cell? All and unwanted and damaged cells are supposed to die. So we phagocytose them away, remove them. So when you say mitosis to repair the cell, you cannot repair the cell. You repair the tissues, right? Let's say you injure already, right? This is your skin, right? You injure, so there's a hole. The tissues here are gone. So mitosis is what? To produce back the cells. They don't repair the cell, and the cell die, go repair and see can repair or not. Okay, this is called resurrection. Put the person in coffin, put under the ground, after that resurrect and then come back. Okay, my goodness. So cell die, they cannot come back. So you don't repair the cell. You repair the tissue by replacing the dead cell with new cells by mitosis. Okay, so this one, today you learn, you score another mark. Every point you learn, you score another mark. So all of you will score A and A star. Can or not? Hello? Why suddenly so quiet? Hello, if can, please type can. If maybe, please type maybe. I don't think I cannot. Okay or not? Okay. Every question score full marks, sure score A star one, right? So your aim is every question score full marks lah. Then you should score A star. Okay, next. Using the term center of the cell. My goodness, what do you mean by center of the cell? Okay, you must use the word equator or metaphase plate when you want to say all the chromosomes align at equator, right? During metaphase. So equator is the word. Don't say center. Where's your waist? Center of my body lah. Center of body where? Center of body. Am I supposed to think of 2-dimensional or 3-dimensional center of the body? Yeah? Okay, my goodness. So this, again, you can see biology, you have to use a term very crystal clear and accurate and specific. Then you score A star. Using the term ends. What do you mean by end? So the sister chromatid separates to opposite pole, not to the two ends of the cell. Okay, the two ends means here and here. You don't go to here and here. Okay, the two ends, here and here. What is the name given here? Pole. Use the word pole. So whatever name that you have not learned up, you still have time to learn up, so you can score full marks for every question, and therefore you score A star. Can or not? Number 5, the mitotic cell cycle. Confusing chromatids. I finished already, no man? Okay, this one finished, sorry. Okay, finished already. Next, number 6. Nucleic acids and protein synthesis. Believing that sugar in ATP is deoxyribose rather than ribose. So ATP is a molecule that you're very commonly known by all of you, which is actually a molecule that provide energy, right? Okay, ATP provide energy. Now ATP is a nucleotide. This nucleotide is a ribonucleic acid. No, it's made up of the sugar. It's made of ribose. Okay, that means it is like a free nucleotide of RNA. How do I remember? I think I remember by ATP is needed in the cytoplasm. Okay, ATP is needed a lot in cytoplasm, in the organelle. So therefore, RNA is also synthesized in the cytoplasm. So it must be RNA monomer. RNA is made of ribose sugar. That's why it's RNA. Ribonucleic acid. Ribonucleic acid, ribose. Okay, full stop. So try to link something related to this so that we can remember, and always remember the correct one. Okay, stating that hydrogen bonds are strong bonds. Hydrogen bonds are weak bonds. Remember, hydrogen bond alone is a weak bond. But when you got a lot of hydrogen bond together, right? So the whole structure become very strong. It's because you got a lot of them together. Okay, so hydrogen alone is a weak bond, right? Covalent bond is actually stronger than hydrogen bond. Disulfide bond is stronger than hydrogen bond. Okay, ionic bond is also stronger than hydrogen bond. So you know the sequence of strongest to the weakest bond, right? Okay, covalent, disulfide is same as covalent because it's covalent bond. Okay, followed by ionic, followed by hydrogen, followed by hydrophobic interaction. This one you must know. Okay, confusing purines and pyrimidine. Now, this one, all of you, I trust all of you memorize already what is purine, what is pyrimidine, right? Okay. Pyramid, right, only has got one top, so it's one ring. That's how I teach my student. Pyramid only got one top, it's triangular, right? It's only one ring. Purine got two rings. Finish. Confusing replication with transcription. Replication is when DNA replicate, right? You make DNA from DNA. Transcription is you make mRNA from DNA. So this one, I think very few of you will confuse. Confusing transcription with translation. No, transcription is from DNA to, type faster. DNA to mRNA, right? Hello, to mRNA is transcription. Hey, what happened? DNA to mRNA is transcription. And then mRNA to polypeptide, not protein, polypeptide. Because protein can made out of a few polypeptide, each polypeptide coded by different gene, different mRNA. Okay, so accurate word is polypeptide, not protein. So you learn how to use the accurate words to guarantee you score full marks, and therefore you score A star. Okay, describing DNA as a genetic code. DNA is not genetic code. Genetic code is the triplet codon, right? The triplet code in DNA, right? The 3 bases coding for one amino acid is called a genetic code. Okay, next, stating that mRNA copies DNA during transcription. What's that? mRNA copy DNA during transcription rather than mRNA is synthesized. Cannot simply interchange one. There's no such thing as mRNA copy DNA. mRNA is synthesized from DNA during transcription. So even when you describe in words, your description must be correct one, you know. Okay, You think of something when you describe out, and when the examiner read your sentence, it must be the correct meaning one, you know. Okay, it must be the correct meaning. Okay. Giving vague molecular definitions of a gene, example, gives you your characteristics. Definition. So now you know that you need to learn up the definition of gene, right? Which is a length of a DNA. That means DNA is so long, isn't it? So this DNA is actually a length or a section of a DNA that code for a polypeptide chain, right? Which is made up of a specific sequences of bases, something like that. Okay, so learn up the definition. Okay, revise again so that you can write your answer. Stating that a gene is a group of 3 bases. Hello, gene is no group of 3 bases, triplet codon, right? Okay, so it's already. Okay, now I'm going to look at common mistakes among students in exam for this chapter, transport in plants. Confusing the roles of xylem and phloem. This one I think you all won't be confused, right? Xylem, this is the name of tissue, isn't it? That transport water and the dissolved mineral ions from the soil to the root to the stem to the leaf, right? Okay, from the root to the stem to the leaf. Phloem is also the name of a tissue. So I think in plant, right, what you need to be careful of is the name of a tissue and name of a cell is different. So when you draw plant drawing, okay, you have to, when you label the structure, you need to label using tissue name, right? So this is what epidermis, cortex, right? This is phloem, cambium, and xylem, right? So you use a tissue name to name those structure, right? But then when you ask to draw the cell, for example, okay, you ask to draw the let's say this is a xylem element, right? Okay, then you have to label this one as xylem element rather than xylem. So the difference between tissue name and cell name, you must learn them up and you must use the right name in the right place, right? And then now they're not saying the name, they're saying the role. The role, oh, you know, like phloem function is translocation, right? Translocation of mainly the sucrose, but also other metabolites, is it? Okay, from the source to the sink. Okay, stating that water moves in xylem by osmosis. No. Water moves in xylem by what? The transpirational pool, right? Which is actually the cohesion tension. So learn up those names. I know all of you know transpiration pool, but in A level, you learn this transpiration pool as the cohesion tension. Cohesion tension, right? Do you know what's cohesion and what's tension? Cohesion, oh, you know, right? Tension is actually negative pressure. Negative pressure means the suction force. That means the transpirational pool that pull the water up, right? And then because of cohesion between water molecules, the whole column, you also need to use the word column of water molecules are being, the whole entire column of water is being pulled up, right, to from the stem to the leaf because of cohesion tension. So this is how water moves, and this leads to mass flow. All these are keywords, right? Mass flow means all the molecules are moving together in the same direction, right? The whole mass flow together. So all these are keywords that you write, you get marks. Right? In biology, you write those keywords. But when you put in sentence, the sentence must make sense, okay? Stating that phloem sap moves by osmosis again, so it's wrong, okay? Phloem sap actually move because of hydrostatic pressure gradient from source to sink, right? Okay, and again, it's also mass flow because the sucrose move together with water all together in the same direction. Okay, next, confusing adhesion with cohesion. This one, I think you all know, right? Cohesion means co means together, same molecules, attraction between two water molecules by hydrogen bond. Adhesion is attraction between the water molecule and the cellulose wall, okay, by forming hydrogen bond, and also with the lignin, because lignin has got a certain component there that is hydrophilic, that the water can form hydrogen bond with. So therefore, the water can be attracted to the cell wall. Which part of cell wall? The cellulose and also the lignin. Even though water cannot pass through, but water can be attracted by. Okay. Anyone want to say, teacher, you prove it lah? Show me the molecule. You want to tell me, okay, I'll show you. Right. Stating, so you can Google search the lignin molecule, and you see that there's actually a part that is hydrophilic, the OH group there. Okay, so that's where the cellulose can form hydrogen bond with the lignin, right? So that causes adhesion. Stating that transpiration is evaporation of water from the surface of a leaf. Oh my God. Oh my God, this is very wrong already. You can see it's very wrong. Evaporation is the moment when the water in the form of liquid becomes water in the form of gas, is evaporation, right? So this is where you cannot simply just write as you wish one. Water in the cell is in the form of, let's say this is a cell in the leaf, is in the form of liquid. After that, when it evaporates in the surface of the mesophyll cell, it becomes gas. So this is called evaporation, right? It doesn't evaporate on the surface of the leaf. In this case here, evaporate on the surface of the mesophyll cell. So this statement is wrong. Unless you're saying that, okay, this is my lower epidermis, and then from the surface, the water from liquid becomes gas here, then yes, it evaporates from the surface of the leaf, yes. So when you describe, the description must be correct. Okay. Transpiration definition is the loss of water vapor from the surfaces of land plant. This is my definition. Okay. Your definition almost close to mine, correct? Okay. Loss of water vapor. Loss of water vapor from surface of land plant, you know. Okay, that means to say this one is not transpiration. This thing that occurs inside the leaf is just evaporation only. It is not transpiration because this water is still inside the intercellular air spaces in the leaf. Let's say here, so these are spongy mesophyll cell, right? The water evaporates into this intercellular air spaces. This is not transpiration because based on definition, it must be loss of water from the surface. This is not the surface yet, it's still inside the plant. So that's why I always tell my student, you want to do well in biology, first thing, you must learn up the definition, because when you learn up definition, your learning and understanding become crystal clear, should or should not, correct or not, you know already. This is not transpiration. It's only when this water vapor in the form of gas passes through the stoma and then, okay, come out to atmosphere. This one is transpiration. So this is transpiration based on the definition. That's why you need to learn up the definition of the biological term. So you know what is what is not what, especially for MCQ question, right? Correct or not? So they draw this diagram and ask you A, B, C, D, or part 1, 2, 3, 4, which are considered as transpiration, can or not? This is part 1, is this transpiration? Part 2, is this transpiration? What about part 3, is this transpiration? Then you must know. So this is how you score as high as possible, and every question you can score full marks. Can, isn't it? Because you learned them already. Actually can one. Okay. Okay, being okay, so of course another place transpiration can occur in the lenticell, right? Okay, lenticell. Lenticell are the opening at the stem of the cell for gaseous exchange, and because of that opening, water vapor can be lost from the surface of this plant through the stem. Okay, so that one is also transpiration. Okay, next, being unclear about definition of a source and a sink. This one your AS trial exam got asked, right? Source and sink got, okay, so this one I no need to say already lah. Okay, number 8. Master, number 8, not explaining closed. When asked to explain closed double circulation. So that means when the question asks you to explain, okay, this closed double circulation, then your explanation must include, what do you mean by closed? What do you mean by double? Okay, for example, I ask you to explain what is fluid mosaic model. Then in your explanation, you must tell me what you mean by fluid and what you mean by mosaic, isn't it? Fluid is when the phospholipid molecules move laterally, right? Fluid. Mosaic is referring to the protein. Protein molecules are scattered throughout the phospholipid bilayer, right? So you have to explain each word. Okay. Close means what? The blood flows within the blood vessel. It's called close. Double means it goes to the heart twice. So therefore involve two circulation, systemic circulation and pulmonary circulation, right? So that's the meaning of you need to explain for each term. Okay. For example, artificial active immunity. Then you must explain what is artificial, what is active, what is immunity. All these three words must explain. Okay. Now confusing cardiac cycle with complete circulation of blood around the body. Huh? Confusing cardiac cycle with complete circulation of blood is different, right? Cardiac cycle means what? Cardiac cycle means all the 4 events that happen in a complete heartbeat, isn't it? In a complete heartbeat. That means to say that the 4 cycle, what are the 4 cycle? Okay, I ask you, okay. Can you tell me what are the four events that happen in one complete cardiac cycle? Michelle, okay, Michelle, what are the 4 events in a complete cardiac cycle? Cepat, Michelle, cepat. Michelle. Hello, faster. Michelle, what are the 4 complete cycles? You don't know, is it? Okay, let's ask another person. Mira Sai. SAN contracts. SAN contracts. Okay, this SAN contracts is talking about the activation of the heartbeat. Okay, activation of heartbeat starts from SAN. But what are the 4 events that occurs in the cardiac cycle? 4 events. Okay, who can tell me? Yes, very good, Cician. Atrial systole, atrial diastole, ventricular systole, ventricular diastole. These are the 4 events in a complete cardiac cycle, right? So therefore, when you study biology, okay, this name called cardiac cycle, what did you learn under cardiac cycle? These 4 steps, which is different from complete circulation of body, right? Which include the systemic circulation and the pulmonary circulation. So which means to say, whatever name given to a process, you need to know what you mean by this name. What process are they referring to? Okay. Now comparing blood with lymph, this one all you know lah. The blood flows in blood vessel. Lymph is a fluid that flows inside the lymph vessel, right? Okay, finish. And then what is tissue fluid? What is blood plasma? Okay, what is the name of the fluid inside the blood vessel? It's called the blood. No, it's not called the blood. The blood is divided into blood cells and blood plasma. So the fluid part is the blood plasma, right? So this you need to know. Next, stating that white blood cells cannot leave the blood. No, all white blood cells can leave the blood because all white blood cells can change shape like amoebic movement, squeeze through the pores in the wall of capillary. So the only cells that can pass through the wall of capillary are the white blood cells. Okay. Confusing carbaminohemoglobin, carboxyhemoglobin, and hemoglobinic acid. Anyone want me to explain this part? Because I think this one is, you know already one. Okay, no need. Anyone? Calling 1 twice by. Okay, next. Stating that carbonic anhydrase catalyzes the dissociation of carbonic acid to hydrogen carbonate ions and hydrogen ions. Carbonic anhydrase, this enzyme found in the red blood cell, is supposed to promote CO2 to dissolve in the water to form CO2 to dissolve in the water to form the carbonic acid, right? Okay. So it does not catalyze the dissociation. After this is formed, it will automatically dissociate to H plus and dissociate to H plus and then HCO3 negative, right? HCO3 negative. And this H plus, because it's acidic, it has to be carried by the hemoglobin. So when hemoglobin carries H plus, it becomes hemoglobinic acid. Where's my hemoglobinic acid? Huh? Here. Okay, right? So probably I need to mention here so they understand better. Now you see that my hemoglobin has got higher affinity for H plus versus oxygen. Okay, what do you mean by higher affinity for H plus than oxygen? Why hemoglobin releases oxygen, it means oxygen is unloaded in the tissue, is because when it comes to the tissue in the red blood cell, because of this process, we've got a lot of H plus in the red blood cell, okay? But hemoglobin is already binding to oxygen. Now, because hemoglobin got higher affinity for H plus, it will throw away the oxygen so that it can come and hold the H plus. So this is the meaning of higher affinity, right? It likes H plus more than oxygen, so therefore it has to throw away oxygen, then only it can hold hydrogen, the H plus. So this is called higher affinity. So that's why oxygen is unloaded and H plus is taken and carried by hemoglobin. So this one leads to Bohr effect. Huh? How do you explain that when CO2 concentration is high, it causes hemoglobin to unload oxygen? Is because of all these processes happening here when you're asked to explain why. Why Bohr effect happen? What is Bohr effect and why does it happen? Bohr effect is when the hemoglobin, we have got lower affinity for oxygen when CO2 concentration is high, so you will unload the oxygen. Why unload? Because of all these series of processes, and finally, the hemoglobin got higher affinity for H plus than oxygen. So throw away the oxygen means unload oxygen, and then it will carry hydrogen. Not hydrogen, sorry, hydrogen ion. Okay. So another thing you must be careful is hydrogen. Okay. Is H or H2 is hydrogen. So the moment you write hydrogen, it's either H or H2. If hydrogen ion is H plus. So you have to use the right word to describe the right thing. Okay. Comparing left-right ventricles when asked to compare left atrium with left ventricle. This one cannot confuse. So you see what the question one, answer what the question one. So you save your time to score more marks in other questions. Don't write something that the question didn't ask. Don't waste your time. Answer what the question asked me. So you make sure you got enough time to answer other questions to score full marks for every question. Okay, this is how you write minimum and score maximum exam. Write minimum, score maximum is your principle. How to write maximum? Just give what they want. Don't write things that they don't want. You don't know how to answer, then you don't know what the question want, go and answer other question. Okay, don't write a lot of things and after that, the whole paragraph got no marks one, waste time only. Okay, go and answer other question. Confusing a description of initiation of heart action with a description of cardiac cycle. Same thing, right? Okay, cardiac cycle, just now we say already, right? Okay, is what is it? Atrial systole, diastole, ventricular systole, diastole, right? Okay. Now, initiation of heart action is just now what you say. Okay, the SAN, right, contracts. Then it sends the excitation wave through the atrial wall, right? After that, it comes to AVN. After that, it goes to bundle of His and then Purkinje fibers, right? Go down the septum and up the heart. Okay, so therefore, the ventricle contracts from bottom upwards. That's called initiation of heart action. So different. So different name, different process. You need to learn up. Gas exchange and smoking. There's no more smoking, okay? Confusing bronchi and bronchiole. So you need to know what is bronchus, what is bronchiole, what is bronchi, plural bronchus and bronchioles. Okay. Describing alveolar wall as cell walls. No, wall is alveolar wall is not cell wall. Okay, so it's different. Stating that ciliated cells are paralyzed. Hello, the ciliated cells originally already cannot move. How can you say it's paralyzed? It is a cilia that can be paralyzed, okay? It's not the cell. The cell originally already cannot move one. Can you tell me what you mean by the cilia is paralyzed? Sorry, the cilia. The cell is paralyzed. That means originally not paralyzed, means can move around the lung. Okay? Some students say, oh, cilia die. How can cilia die? Cilia got no life, only cell can die. So this is using the right word and right context. So don't simply write one, okay? You simply write, let me say what? You go pasar malam, you call everybody and you ask about bio, they tell you like this. Then no need to study bio, okay? Give me A-level standard bio. Don't give me pasar malam. Pasar malam bio, no, okay? Right. Don't give me layman term bio, okay? You're sitting for A-level exam. Give me A-level standard bio. Okay, next. Stating that lung cancer is no need COPD. This one no need to. This one all no need. This one all no need. Why no need? Because not in your syllabus. Okay, finish. Next, infectious disease. Naming an organism when asked what type of organism. Type of organism causing the disease. So what type? Is it a bacterium? A bacterium? Is it a virus? Is it a prototype? Is type of organism. So name organism versus name the type of organism different. Read every single word in the question, please. Because you want to score full marks for every question. Full marks. Are you stressed? I feel very stressed. When I say full marks, are you stressed or not? You can see it's actually very easy to score full marks. Read carefully, okay? Stating that Lo Wen Tao is very stressed. Every time I say every question must score full marks, I get stressed, you also get stressed. But actually it's possible. Okay, you have to train yourself until you will work so hard that every question you try to score full marks. That's where you can score A star and not only A star. I don't want to say it after you get even more stressed. Okay, enough. A star is enough. Okay. Stating that malaria and TB are caused by viruses. Ayo, this one, how can? Malaria is prototype, right? TB is what? Bacteria, isn't it? This one confused me. Please go and study properly. Confusing HR. This is what happened in your AS trial exam lah. Right? HIV is referred to the virus. Right? The organism that eventually will lead to the disease. Okay, the illness. I don't want to say the disease. The illness after HIV infection eventually will develop into HIV/AIDS. AIDS is a collection of opportunistic disease because of HIV. Okay, HIV leads to immunodeficiency, which eventually leads to a lot of opportunistic diseases. These opportunistic diseases, this group is called AIDS. This group is called AIDS. So is this infectious? Not necessarily. AIDS is not necessarily infectious. It depends. If it's caused by pathogen like TB, yes. TB, yes. But can you tell me skin cancer is transmissible, not skin cancer because of AIDS? No, right? So you cannot say AIDS is transmissible, is infectious. No. Some of them yes, some of them no. So this called, you need to learn to that specific detail so your answer is accurate and crystal clear. Then you can score full marks. Accurate, that means perfectly correct and crystal clear. Then you can score full marks. Okay, cannot, actually can. This is how I do my bio one. This is how I study my bio. Every question must score full marks. Every question must score full marks. Then when you study, you study and pay attention to all the detail. Why is it not possible? It's possible. Okay. I mean, for your case, it's possible because you're willing to work hard. Willingness to work hard definitely can score very high. Correct or not? Unless you say, this one too difficult, I don't want to study. This one I don't want to study. Then too bad lah. Okay, no reason cannot score. Stating that antibiotics cannot work on viral cell wall. Virus got cell wall, man. Virus got no cell wall. That's the problem. Virus got protein code. So antibiotics cannot work on viral protein code, correct? This is a common mistake among students. You keep on appearing again and again here, right? Virus got no cell wall, only plant cell and bacteria cell got cell wall, full stop, enough, no virus. Using the term immune instead of resistant, this is where students might actually use these two terms interchangeably. I already said in biology, each word got their own meaning, cannot use interchangeably unless you go pasar malam. Okay, immune means immune what? Immune in our bodies involve the phagocyte, that phagocytose, right? After that, you got the T helper cell, right? Clonal selection, clonal expansion. Then after that, it will go and secrete the cytokine or the lymphokine, right? And this cytokine, lymphokine will go and activate what cell? What cell can be activated by this cytokine? Either B cells, the selected B cells, if you want to secrete antibody, or the selected T killer cell, TK, also called T cytotoxic cells, okay, that will go and kill the 3 types of cells in the body. So when you involve these cells here, it's called immune response. Immune response. Okay, it's not called resistant. Resistant is when you say, okay, the body is resistant to a certain chemical. Okay, for example, bacteria. Bacteria become resistant to the antibiotic that we use. Okay, the mosquito, Anopheles mosquito become resistant to the insecticide that we use. It's called resistant to. No immunity involved. So you cannot say, oh, bacteria immune to antibiotic. Or bacteria got phagocyte, T helper cell, B cells, is it? No, isn't it? So when you learn, this is the word you learn for what context, only used for that, cannot simply interchange. Then you can score very high mark in exam. Can or not? Actually can. Miracles will happen because of that. I feel very stressed already. Okay, you feel stressed or not? Never mind, just try to score full marks for every question. Using the term immune, okay, finish. Lastly, okay, last already, right? Very happy, last already. Okay, last. Confusing the roles of B and T cell. This one means you go and learn up lah. What is the role of B cells and T cells? Just go and learn up. Okay. If you need me to explain, tell me. I'll explain everything. Just now, I explained almost everything. Okay. Stating that a pathogen is an antigen. No, okay, pathogen is not antigen. Pathogen, right? For example, this is a bacterium, right? Okay, this is the virus. Okay, I draw this HIV virus. Okay, because the virus got membrane, right? And then got the protein coat, right? Okay, and then you got the DNA inside here, whatever. So this is the pathogen. This bacteria, like I said, Mycobacterium tuberculosis is a pathogen. This virus HIV is a pathogen. Okay, so this is a bacteria. It's a pathogen. This virus is a pathogen. Pathogen must be an organism. And in this organism, just like all the cells that we've learned, the cells got their own name tag, right? So the name tag is called the antigen. So this bacteria cell wall has got its name tag. So if I draw all circles, means all of them are the same type of name tag. Sometimes you got a few. Let's say I draw this triangular, it's a different type of name tag already, right? Okay. So this one, you got the protein, isn't it? Which act as an antigen on the surface, membrane of this HIV virus. Right or not? So all these are the antigen. So the antigen is not the pathogen is not the antigen. The hair is not you, you are not the hair. It's the same thing. Is the hair you, or you are the hair? Which one is correct? Lo Wen Tao? Is the hair you, or are you the hair? Lo Wen Tao. It's the same thing, right? Lo Wen Tao got no response. Yes, I'm the hair. You are not the hair. When you wash your hair after you shower, the hair on the toilet, is it you? No, it's not you anymore. So the hair is not you and you're not the hair. Okay, finish, full stop. So the pathogen is the antigen, not pathogen. Finish. Okay, so this is called learning the detail using the right term. To that detail, you learn to that detail, you have to say. Okay, next one. Stating that a memory cell remembers. Ayo, memory cell do not remember lah. Okay, cannot simply say this called pasar malam answer. How to get the mark? You need to give A-level answer, not pasar malam answer. Remember means pasar malam. Memory cells can remember lah. Everybody can say that. Then what for you study A-level? So what does the memory cells do? Long-term immunity, right? So the first thing you need to say, memory cells, you use back the biological term that you have learned related to this. It's called long-term immunity. This 1 mark in exam already. Finish. Give the name 1 mark. Long-term immunity. Okay, tell me what you mean by long-term immunity. So that means to say that when this person is exposed to the same antigen in subsequent infection, or subsequent exposure, okay, subsequent exposure. That means after you expose to the antigen for the first time, in your subsequent exposure, you already got memory cells. So in a subsequent exposure of the same antigen, then your memory cells, right, will be activated to do what? Activated to do what? Okay, that means it will divide, it will differentiate to more, what? More plasma cells and memory cells, right? And then what does the plasma cells do? Plasma cells will secrete antibody, right? And then what the antibody do? Bind to antigen. So this is what does the memory cells do? You give me a term, long-term immunity. Then exactly do what? Give me all the detail. So this is how you write your answer in exam. Because you write long-term immunity, 3 words, you get 1 mark already, you know. Why not write this one? Always write the name first. Name of structure, name of process, already 1 mark. 3 words only. Then the sentence is always longer. So give me the name first. Then you elaborate. This is how you write minimum and score maximum. Strategize. Lowenthal, are you stressed? Confusing antigen binding sites on antibody with receptor. You learn that receptors are found on the membrane, right? This is for the signal molecule like the hormone to bind, right? This is where you learn receptor. Then when you learn the antibody, all right? So antibody, you can be asked to draw the structure antibody. You make sure you learn how to draw. Am I drawing correctly? Where's my second one? Is it here? I think it's like this. Okay. So the antigen binding site is actually here, right? This 2 part is called the antigen binding site. So you cannot interchange the name of the antigen binding site to become receptor or active site. Active site is for what? Enzyme. Receptor is for what? In the membrane, right? Antigen binding site is for what? The antibody. So cannot simply change the name, okay? Can I say that Lowenthal got a lot of antenna? So when the Lowenthal shower, all the antenna drop down to the floor. Or all the cilia drop down, all flagella drop down. Cannot, right? Hair is not flagella. Same thing here. I think so. Lowenthal, after you go swimming pool, right? All your antenna drop down the swimming pool. Poor Lowenthal. Yes, you are the head. Lowenthal. Now you know you can laugh. Then you can see all sorts of answers the student write. That's why after I mark your trial exam paper, my hair, right, from like this, right, very nice, all stand up already. After that exploded. No more hair. When I read all sorts of answers. Okay, so now you know how to score full marks, right? Don't interchange the word. What you learn in your syllabus, use back the same word, okay? Stating that antibodies are mixed with myeloma cells to form hybrid. I actually saw this kind of answer, right? Wait, in your AS trial, you got this question, right? Antibodies are mixed with myeloma cells. No, how to produce what? Hybridoma cells. You have to fuse 2 cells together, right? So how can you fuse antibody with this one? Antibody is not a cell. So you're supposed to fuse plasma cells with myeloma cell, which is a cancer cell of bone marrow. Myelo means bone marrow. Cancer cell, bone marrow, to form the hybridoma cell, right? Okay, so this is when You're not very familiar yet. So now you work on familiarizing yourself with everything in your syllabus. Whichever part that you have not been familiarized with it. Then you can score full marks for every question. Using the name of a disease to mean the actual causative feature, same thing lah. So when the question asks you to give me the name, what are the 4 diseases you've learned? Okay, I've got the cholera, right? I've got the TB, tuberculosis. I've got the malaria. I've got the HIV/AIDS. So the name of the disease is different from the name of the pathogen. So they ask, so therefore give the correct one they give. Can or not? Okay, any question? If not, we go for a break.